Hello everyone. I'm going to talk about some work on wheat I did over the summer with Dr. Swarbrick, who's a fellow here. So carrying on the climate theme, which we've had so far, there's a lot of interest in designing better forms of agriculture in terms of reducing carbon emissions. One of these is regenerative agriculture, which is a bit of a buzzword, but broadly we can characterize it by about 5 pillars. So low tillage, or aiming not to plough the soil as much, if at all. Using cover crops, so other crops in between, say wheat, to cover the soil. In linking to the first point, we want to keep the soil intact as much as possible. And the aim of this is to preserve networks of fungal mycelia that might be linking plant roots as well as the plant roots themselves from damage. And we also want to aim to plant genetically diverse crops. That's advantageous from a disease-resistant perspective because monocultures are particularly susceptible to epidemics tearing through them. And we would also like to have living livestock on arable land. Not only is that a better use of space, but there's some potential for fertilizer use and bringing down costs of agriculture. So these 5 points will potentially be a way to increase biodiversity at farming locations, reduce use of machinery, and potentially reduce carbon emissions as well. But the problem with having different sorts of agriculture is that our current crops are adapted for certain types of agricultural practices that are not exactly like this, more high tillage practices. So one of the aims of my project was to actually look into some lines of wheat, synthetic wheats to be precise, and to see whether they were better adapted to regenerative agriculture. So what is actually a synthetic wheat? Well, when modern wheat as we know it today, when it was domesticated many years ago, there was 2 hybridization events. Both occurred by chance, and they produced a rather complex hexaploid genome. You can see in the bottom left-hand side of that figure. And the problem with those hybridization events is that they both captured only a sort of a snapshot and a micro snapshot of the existing genetic diversity that was present in the wild. So modern wheats today are rather undiverse and probably not very representative of the genetic diversity that exists in the wild. That's then a limitation when it comes to producing new varieties of wheat. So the idea of a synthetic hexaploid wheat is to recreate essentially ancestral hybridization events, but aim to capture more diversity and do it in a more intelligent way, to look at the lines we're then producing and then carry some forward for developing as agricultural lines. So NIAB, which is just down the road, has created several lines of synthetic hexaploid wheat, and that form the basis of the genetic resources which I worked with. So these are the synthetic hexaploids, and they were used as donors to cross with a sort of a background parent, which is Rokos, which is an elite. Well, it's not so elite now because it's kind of out of fashion in the wheat world. But Rokos is essentially a store of reliable genes, and Rokos was repeatedly crossed with these primary synthetics, so that we get a balance between incorporating novel diversity from our synthetic donors and potentially better adapting them, say, for regenerative agriculture, and Rokos providing just a strong support and known characteristics. So that produces a bunch of different lines, of which some of them, these selection lines that you can see in the bottom left-hand corner, were selected for being especially high yielding. So some of the work I did was characterizing these lines based on arrays of genetic markers. It's too expensive to sequence the vast numbers of lines that are produced, say, at NIAB. So instead, we opt for a slightly simpler approach, which is just to look at certain nucleotides and then use those as a proxy for the whole genome. So here is a map of Rokos we can see as the background parent, and this was crossed with the primary synthetics to produce the offspring lines here. And then this enables us to see which offspring lines are most closely related to one another. And I did that genome by genome. So wheat has A, B, and D genomes. So if we break them down into A, B, and D, then we can uncover more detail than we might see at the whole genome level. Linked with this work is consideration of strigolactone, which is a plant growth regulator that Dr. Swarbrick works on. And there's a set of genes involved in strigolactone synthesis that we know of and involved in its perception. One of those, for example, is D14, and we know it's critical for the sensitivity to strigolactone because if we mutate it, then we get rice with accelerated shoot growth. So this set of genes can therefore form a basis for a construction of a set of genes of interest, which might change yield under regenerative agriculture. So part of my work was to look at these genes and see whether they are present or introgressed in the offspring selection lines. And this is a graphical genotype map that shows us which regions of DNA are from which parent, essentially. So Robicus is our reliable background. This is almost all yellow. And then down here are the primary synthetic hexaploids with a lot of, we can see this vast variation in color is due to the extensive genetic diversity. And then in these lines here, which are the offspring lines, we can see that some of them have incorporated more genetic diversity than others. This, for example, has not incorporated very much genetic diversity from the primary synthetics, whereas 222.8.8, the top one, has got big introgressions, chromosomes 3, 5, and 7 in particular. So zooming in on that, we can actually look kind of manually at the D genome of this, and there are 7 chromosomes on the D genome, and we can classify the introgressions based on whether they actually occur in a gene of interest, so, or rather, if the gene of interest falls within an introgressed region. So here we can see on 5, the big introgressed region on 5D has a gene of interest introgressed. Similarly, 3D as well, and 7D. So that's indicative of some potential phenotypic differences that might be interesting to look at further in a pre-breeding program. The other strand of my work was to do with physiology more underground in the soil, and working on a data set that had been partially collected, looking at the effect of regenerative agricultural conditions on how much fungi are colonizing the roots. So plants do actually obtain quite a lot of their nutrients in many cases from symbiotic fungi that will supply, say, nitrogen or phosphorus. So if you want to apply less fertilizer, for instance, fertilizer isn't a renewable resource, we might want to encourage fungal colonization, but does regenerative agriculture actually aid that or not? It's a key research question. It's one that we tried to address here. So we systematically altered level of tillage, so how much you're plowing the soil, how much cover crop there is, and how much nitrogen is applied. And then people from the Crop Science Center went and took some roots from each of these plots, washed them, dried them, and then we found that most changes in agricultural conditions didn't produce significant results, at least not with the material we had, except for when we varied both nitrogen level and tillage level. We seem to see some association with that on the presence of what we call extra-radical arbuscular mycorrhizal hyphae, which are fungal threads that poke out from roots. And these are associated with the interchange of nitrogen from the soil going into the roots. And they're quite rare, so it's possible that we maybe saw this effect because it's particularly easy to spot them. You really can't miss them, and there's also a sensitivity. So there are certain plants we saw in the sample data where there weren't many, and at other points there were loads. Whereas with some varieties of fungi, we looked at several, including things called fine endophytes as well as classical AMF. Several, you just see them all the time, so they don't display that level of sensitivity. However, we did only use quite a small sample size because all of these things have to be scored manually. So for each type of fungus, we looked at about 100 fields of view per root, and that was done by humans to sort of classify whether fungus was there or not, which is obviously difficult to do, prone to error, and means that you can't really go very fast. So there's a lot of subjectivity, as I said. In this picture, for example, is a picture of fine endophytes colonizing a wheat root, one of the roots in the data set. We can see this sort of fine network that is visible, but there is a lot of interest in further characterizing this diversity. So one of the people I worked with at the Crop Science Center is going to sequence the roots in the data set, so that will provide a better picture of actually what fungi are involved in this symbiotic relationship. And also leveraging machine learning, which I think you'll hear about a bit later, to do this classification. So would it be possible to train an advanced model to not only spot where there are fungi, but also classify these fungi into the several different categories that do exist, because this would really increase the throughput and actually enable us to get much better statistical power to actually properly answer these research questions about regenerative agriculture. So a big thank you to Stephanie, who supervised my project, and also to Chris Rokos for supporting. Thank you.